Good evening, everyone. Today we will be talking about the chromosomal aberration. Chromosomal aberration. So, first thing is what is chromosomal aberration? Like if there are change in the genes, and because of that change in the genes, any any defect or any syndrome can happen to a child. So this has happened because of the gene change. It can be of two types basically. That is numerical, and second is structural. Now, what is numerical chromosomal aberration? In numerical chromosomal aberration, it is basically there are change in the gene number in the chromosome. So it can be happen like the whole chromosome has moved from one place to an another place. There are 23 genes in a chromosome, so it can be possible that whole together 23 genes has came. So this 23 has become 46 or 69. So this is a multiple of this will be a multiple of 23. So that is called euploidy. So if uh, uh, if the total number of genes in a chromosome, if it is a three times of the 23, then it will be called triploid. That is 3 into 23. And if total number of chromosomes is four times, then it will be called tetraploid. That is four times into 23. So this is euploidy, which happens because of the n times of number of genes, that is 23. And now the second case, which is aneuploidy. In aneuploidy, what happens? The whole means all the genes in the chromosome will not shift to the second chromosome. Only some genes will be transferred, like one genes or two genes, like that. If suppose we know that in any cell there is total number of 46 chromosomes. In that 46 chromosomes, if one gene has came extra, now this 46 will become 47. In that case it is called trisomy. If total number of genes has become 47, we write like plus 18. This is Edward syndrome. Second case is monosomy. This is trisomy. Monosomy, in what happened, like the total number of genes are in the chromosome is 46. If one gene will go somewhere else, like if one gene got separated from the total number of genes, and the final amount of genes, the resultant amount of gene, if it is 45, then it is monosomy. This can lead to Turner syndrome. Turner syndrome. So this is about numerical chromosomal aberration. Now, there are various disease which can happen due to this, like Edward syndrome, Down syndrome, with Down syndrome and Edward syndrome, like Turner syndrome, which is because of the change in the gene number in the allosomal cell. And what is allosomal cell? That is the sex cell. Then clean filter syndrome, that also happened because of this numerical aberration only. So this is numerical aberration. Now what is structural chromosomal aberration? So what happens in the structural chromosomal aberration? Like the number of genes will not change. In a same gene only, the some part of the gene will be got deleted or duplicate or inversion, translocation or ring chromosome. So what happens in the deleted? Some part will got deleted and the means now it will have effectively dead genes only after the deletion. Second is duplicate duplication. As the name suggests, some part of the genes will duplicate itself, and there are two copies in the chromosome. Then third is inversion. So in inversion, what happens basically like uh, like this. Uh, this is our chromosome. I will make it here. This is a chromosome. Suppose here it's some gene, and here it is some gene, and if it becomes like this, BA, this is inversion. Then translocation. Translocation means rearrangement of part of the chromosome between two non-homologous chromosomes. It can be balanced, even exchange between two non-homologous chromosomes. This is one chromosome. This is second chromosome. This is echnocentric, like echnocentric, and these both, if these both the chromosome will merge with each other, it will be like this. And that short arm will be get lost, and this part got merged with each other. So this is translocation. So if a person having this type of genetic defect, that genetic translocation structural aberration, then that person will show a characters that is similar to Down syndrome. Then ring chromosome. What is ring chromosome? Like suppose this is a chromosome and this part, the top part and the bottom part. If it got deleted, it will form a ring after this, like this. This part get merged with this part. It will form like a ring structure. This is called ring chromosome. So this is about the structural aberration. That is the end as of now. Thank you.